Well, thanks for the invitation. I'm not going to talk about anything from prior speakers, so hopefully sets a good stage for what this is. I want to first start, besides thanking the organizers for inviting me, and thank you for listening. I also want to thank our partners on the project, Science Exchange, who's been working with us to help conduct this project over the past couple of years, as well as obviously not going to be possible without our funders, Arnold Ventures, who actually funded us to be able to do this work. Let me give a little bit of background myself, too, that'll make sense, I think, for the title. So I have my PhD in biomedical research with a focus on preclinical cancer biology. And so I've been working on this project since 2013. And one of the things that struck me, and right before I started this project, before I joined the Center for Open Science, were these really kind of bombshell papers from pharma in 2011, 2012, basically indicating that they could not replicate preclinical work. That in the course of their evaluation of whether it's worth continuing to invest in markers or drug targets for a potential avenue towards getting that into a clinical trial to eventually get it to market, that they failed more than they would have thought. And obviously you can imagine there's a lot of finger pointing that could potentially come off in that. But one of the things that really struck me, and I think struck a lot of people that maybe we don't talk enough about, is that what we highlight right there is all that they said. They said that they couldn't replicate the papers. So we don't know what papers those were. We didn't know what experiments they were. We didn't know what results they found. We didn't know what they meant by replicate. And there were reasons for that they share, right? Non-disclosure agreements. This is part of also not something that they went out to do. It's part of their routine business. So part of not disclosing this would also be not disclosing essentially where they're looking or where they were looking in terms of drug targets themselves. But nonetheless, this is used constantly. These papers are used constantly as an indicator that there is, a replication problem. But there's no evidence, right? There's no evidence behind it. Just like we can easily make a research claim, but it's a lot more important to be able to show that evidence behind it. At the same time, so basically, let me pause for a second. When I started, we basically were saying, well, great, let's do an open project. Let's see if we can do the same thing. Let's replicate the results. Let's do it in the open. And let's actually understand, like, what are we finding, right, when we conduct replications. At the same time, the reproducibility project in psychology was ongoing, and it was basically used as inspiration for our project. So we wanted to basically take everything that project was doing and did and use it as an opportunity to piggyback off of it, right? Leverage what made a lot of sense. So things such as, let's do a systematic selection. Let's not do some biased selection based on what are viable drug targets. Let's actually kind of understand what's out there, similar to what RPP did. Let's pre-register. We know those are important approaches to increase the quality of the design, so we want to take that on as well in this project. Make sure it's all open from the beginning to the end, and don't hide anything, so that way we allow all the evidence to be able to be evaluated by anybody who wants to look at it, opposed to just making a claim. And importantly, we wanted to engage the authors. This wasn't meant to be combative. I still believe this today, which is when you conduct a project like this and you do it in the open, you're going to win. Right? You're going to find out what works, you're going to figure out how you can amplify that, and you're going to figure out where the challenges are, and you're going to figure out how to improve that. Right? It's just holding that mirror up. It's not saying you're right, I'm wrong. It's saying, what are we doing? Because we don't really have an idea until we kind of do a project like this. And part of that is to engage the authors, not be combative with that. So RPP did that, and we wanted to mirror that as much as we could in our own project. We also wanted to raise the game a little bit, right? Things had changed. The psychology project started, I think, 2010, around 11. And it moves rapidly. In fact, if I had to go back and do it today, this would probably still be way out of date. But at the time in 2013, 14, when we were launching this, we wanted to take, kind of, what else can we add to the approach that we're using? And the big change that we did was registered reports. There was a new format that came out that we knew that could increase the quality of the design, also allow us to have kind of a third party interact with us and the authors in terms of making sure that we're doing quality designs. And we found partners in that in the journal eLife. So briefly, kind of large view is that we identified 51 papers that were high impact in preclinical research. I'll talk a little bit more into how we did that in a moment. We made everything open. That's the site on OSF. It's continually being more and more open. Every time we finish a project and publish it, we open up everything that's behind it. And one of the things that's unique with eLife and the registered reports format that worked well for us is early in the process, right, we're gathering informal feedback with the authors, trying to figure out what is the best design. And when we send it off for peer review, eLife didn't want to exclude the authors, the original authors. So an original author was included in that peer review process. And that's a way for them to have a voice. At the same note, they also wanted to have experts in the field and a statistician, right? Because obviously we want to make sure that the sample sizes that we're choosing and the statistical analysis that we're doing is robust and appropriate. So similar to any other registry reports format, we have 2 stages. First stage is pre-commitment by us as researchers, the reviewers, the editor. And then the second stage is to publish those results, regardless of outcome, as long as we adhere to what we plan to do beforehand. And similar, oh, it's getting cut off on the bottom. Similar to RPP, we wanted to analyze. The main goal here was to say, well, I want to be able to compare outcomes of replications to outcomes of the original and be able to do a meta-analysis on that. I'm not going to show that today, so quickly set yourself. That's not where we're going. Instead, I want to talk about what we were doing, the process, what actually happened as we did this project over the course of the last 5+ years. All right, so as you can see, I'm going to kind of walk through a little bit, knowing that I get the advantage now in hindsight to be able to kind of coalesce this all together. It's not as if it actually portrayed where we naturally walk through each step at a time, but it does allow us to kind of get some aggregate results. All right, so where did our papers come? I told you we started in 2013, so we went back 3 years. We said 2010, 11, 12. We wanted to know, find identified papers using different search terms in Web of Science and Scopius, so words like cancer, onco, and the such. And we basically were hunting for cancer biology preclinical study papers. So besides excluding clinical trials and reviews, we also excluded certain types of papers that were wholly genomic, proteomic, high throughput based, basically exploratory approaches, right? We were more interested in that kind of confirmatory research, what's followed up after you find some really interesting hit. It doesn't mean that we excluded papers that included those. It means that those were not part of what we were interested in studying. Partly because there's different challenges, but also because they're used largely as the basis to generate the hypotheses that get tested in animals and in cell lines or with patient samples. We selected based on impact. So we used citation and readership at this point, Mendeley counts, right? Who was reading the papers and who was reading the most ones, and basically ranked them, and we picked the ones from the top. So I guess surprisingly, not surprisingly, Nature, Science, Cell comes flying out at us. That's the majority of the papers we had. There are a couple others, PNAS and genomic research as well. So this was essentially our sampling frame. Of note, it's also common in preclinical cancer research and biomedical in general, not always to show all the data, right? We do a lot of representative images. So there's a lot of images in certain techniques like Western blot, where we'll show one result, even though we've done it multiple times. And that's important because that allows us to be able to understand the fact that there are multiple experiments and there's different ways to represent those experiments. And also common, especially in these journals, is dozens and dozens of experiments. A couple journals maybe had 3 or 4. The Nature ones on typically would have close to 50 different experiments. Basically the supplement itself was like 5, 6 papers. We were not going to do all that. We would have sunk all of our resources doing a fraction of a Nature paper. Instead, we decided, let's just pick the key experiments, the ones that embody that main conclusion. That's obviously a bit subjective, so again, peer review helps us a lot in terms of identifying that. But I'm going to give you a quick overview of kind of what we found in this design stage. So quickly kind of hit where we started. So we started with 51, like I said. Of that, 197 experiments, that nested structure, around 4 experiments on average we selected with a range. When we finished this phase, we went down to 36 and 114 experiments. So now I'm going to tell you how did we kind of shrink down in terms of our scope? What did we face that made us do this? So one way to start doing this is to think through the questions that we had to answer as we were conducting this project. So one of the first things we want to do is conduct high-powered replications. So it means I need to understand what the original data was. I need to be able to look at that, get a good estimate of the effect size so I can properly say how many samples do I need to have. That means I need data, right? I can do it, but I'd have to make some massive assumptions. So we asked, in this case, we're saying, for the experiments we selected, were there any papers that had all the data open, either in the paper or in a repository? And there were none of them. And in fact, of the 197 experiments, only 3 of them had it, that had open data. When we kind of break that down a little bit more, we can look at, sorry, let me back up a second. Because we didn't have the data, we went to ask the authors for the data, right? And so then we can start to dissect that a little bit more, right? So if it's obviously open, that's easy. I don't have to talk to the author to get the data. But if it's not open, I want to ask the author, can you share the data, right? So representative images, can you share me the rest so I can start to understand the variance of it? A bar graph, wonderful. What is the data that's behind that bar graph? What can you share with me that's not in that paper? What we found is that for the experiments, we'll start there, is that the vast majority couldn't share the data. It wasn't available to them, they couldn't run it down, or they weren't willing to share it with us. Like I said, there were 2% of the experiments that actually had open data. And when we got data, it would either be the raw data, which we were hunting for and hoping for, or it would just be the summary data. They would say, well, here's the means and the standard deviations that we use to plot the graph, which means that we had to make assumptions of what the underlying distribution was. And so we can also translate that back out to the papers. I'm going to do this back and forth between experiments and papers because of this nested structure. So if we go back to the papers, let's be a little more liberal. Let's ask the question, well, what if of any of the experiments that we selected in the paper, they had, say, open data, or they shared the raw data for one of, say, 5 experiments. So if we take that pretty liberal definition, what you see here is that still over half the papers didn't share anything for any of the experiments. So this is a challenge, which means that we had to make assumptions, right? We had to either estimate the data from the graphs and assume underlying distribution, or we'd have to come up with another strategy to be able to say, well, how many samples should we do to conduct a rigorous and high-quality replication? Similarly, just like data, I need to know what to do. We need to know what we're going to actually conduct. And so naturally, we have material and methods sections in papers. And so what we needed to appreciate was, well, in order for us to be able to conduct replications, I need to know what you did. I need to understand the methodology that you used. I'm going to take a note here real quick, because I know that we have a diverse audience. Something that needs to change, but nonetheless is still commonplace in a lot of journals, is that we have dozens of experiments, yet when we get to the methods sections, we lump it all together as if they all counted for the same thing. So I used cell lines multiple different ways, but I'll lump it together and tell you all about cell lines, or I'll tell you all about a mouse model, even though there's 10 of them. And so there's a disconnect, this massive disconnect, this hoop I've got to jump through to say, well, what did you do for that experiment? Because I got to now figure out what bits and pieces you used across the material methods section. So naturally, when we finished this, we knew we needed information from the authors. Now, one, that makes sense because more information is always better. But again, we can break this down a little bit. When we figured out from the papers for these experiments, what did we need, we can kind of vary them across the degree, which is anywhere from few clarifications to these extreme clarifications. So what does that mean? Let me give you a little bit of context to see if I can try to communicate this both to biomedical researchers and not. We'll start with the extreme. So I'll get this experiment, and an example of this we'd have, where the original study is looking for the efficacy of the drug on tumorigenesis in mice. Okay, so I would hope that they give me enough details to figure that out. But an extreme clarification would say, I knew you used the drug and you told me the drug, but I don't know what dose you used it in. I don't know what strain of mice you used it in, let alone the sex of the mice and the age of the mice. I don't know how long you treated it for. I have a guess what your endpoint is based on your graph, but I have no idea how you calculated it. So I can do this replication, but I have to make extreme assumptions of what's going to go on there. I don't even understand what you did, let alone attempting to do the same thing with any fidelity. Versus a few clarifications, which take that same experiment, somebody explains to me, okay, well, this is the strain we use, this is the sex of the mice, this is the age of the mice, or this is the drug. Maybe they leave out what the vehicle they used to put the drug in, or the exact way that they injected it into the mice. I know those are factors that are really important, but they're vastly different from the other one that was an extreme. And this doesn't even touch upon all the other factors that we either don't know because it wasn't disclosed that could be at play, or all the other mundane instances that aren't mundane until they become important for us to know. So such things as like the really exact conditions of what your incubator temperature is. We know that's important. We didn't even get to that. So this is still a fairly liberal definition of what clarification means. All right, so naturally we didn't stop there. We went, great, hey authors, can you help us? Can you help us understand what to do? Here's what we found, here's our questions. Obviously there's probably more questions. What insight can you help with us? And what we found was a bimodal response. We'd either have some people that were just not helpful at all. We found some people that were really amazingly helpful. So again, I'll give you an example of not helpful at all. One would be, leave me alone, figure it out for yourself, the paper tells you everything. Okay, that's not very helpful. I can do that, but it's not really helping me. Actually, and similar to that, which is, just use standard methods. It's all good. You can use standard methods. Okay, great. That's, again, I didn't need to email you for that. Extremely helpful would be, I'm going to go talk and hunt down all my authors. They've left the lab, but I'm going to go find all of them. I'm going to share what you had. I want to get all their insights. I'm going to have every single one of them who are relevant to the experiments you're interested weigh in. And they're hunting them down all over the world, right? Because the research, the instant it's published, everybody's gone and left. So to me, that's extremely helpful. And they wouldn't just answer our questions. They'd tell us all the other things that they knew. They'd share us their protocols, right? Detailed steps of exactly what we should do. To me, that was extremely helpful, really, really useful. So you might be asking, which is a fair question because we asked it, well, is it just because they're really not helpful because you asked too much? But in the instances where you didn't ask very much, that's when they were extremely helpful. So if we look at the papers and we kind of plot that, no, it doesn't. It's kind of all over the place, right? That we see that just as equally as there are authors who are helpful in terms of the degree of what we ask, we see ones that are just as unhelpful in that same exact range. So there's still something else that's at play there, but we still see this kind of varying response of authors based on what we were asking for. All right, so I'm gonna pause for a second and kind of, I think I'm painting a bit of a picture, but it's gonna get a little more complex. So we'll see if I can kind of express this, which is we ran into challenges. That's basically the message of this entire talk is. And we ran into these issues, getting the original materials, which I haven't talked about, but there are some examples there I can give you in a second. Maybe we have to recreate them, designing it, trying to get information that we can, or making assumptions. And there's all of these changes that are going on. This is not unlike typical research, right? I've got all of these constraints, time, budget, resource allocation. In a typical environment, you'd also be assessing the viability of the experiments that you're doing. Is this heading in the right direction? Am I going to actually get, say, a paper out of this, or is this a viable solution? How much evidence do I have? Like, how confident am I in the evidence that I've collected or not? Is this should I stop this whole line of study or should I keep going? We were less interested in that. We were very dedicated to we're going to run this thing through no matter what. But the first ones, the ones of the resource time challenges, was very paramount to us. We had a lot of those, right? We were doing multiple replications. We were trying to manage getting materials from different institutes. So material transfer agreements was a big time suck for what we were doing. The science exchange, sourcing multiple labs throughout all of these experiments. We're trying to get all this information exchange from the authors, the reviewers, to update what we're doing. All this coordination that's going on was massive. And I think the easiest way, instead of kind of dissecting all of that, is just to show you cost, the bottom line. What did this cost us? And how did that change over the course of the experiment, the course of the project at whole? So to give you kind of a little price, what I mean here is we started when we pitched this project naively was, it's only going to cost us $25,000 to do an experiment, so do a paper replication. Not quite defining everything that we did, but we thought that was a viable option to throw out there. By the time we got to stage 1 review, which is what we said, we're all ready, hey, eLife, we're going to send this to you. We massively watched that go up because of these challenges that we had, just interfacing with authors and getting information or not getting information. By the time we finished peer review, rightly so, they asked us to do sometimes more, right? Sometimes they switched things. They said, no, that's not the right. You need to do an extra step, some more quality control, or do something else to make sure that this is the right way to conduct this experiment. So that's good. That's why we're using that format. And naturally, there's a consequence, which is it's going to cost a little more. And then you'd think, okay, great, you guys did all this, just go run and do it. No, the costs go up again. And we'll get into that in a little bit, which basically means that more than double the price increased in this one aspect to kind of illustrate the challenges we had. And what that meant while we were doing this, and I'll give you an example from materials, is we were constantly struggling with what to do. So we were getting insight. It's not like we started this all at once. As papers were moving through the process, as they were starting to finish, we got wind of the challenges and the impact it was having on our time and costs. So that means we had to make adjustments about the ones that were earlier in the process. A good example of kind of some of the tough decisions we had was, well, what should we do if they don't share key reagent materials that are not publicly available in any way? So do we remake it? If we do, that's going to cost more time and money. Or do we get rid of the study? Well, we did both. We had those situations. We had one where the authors refused to share their mouse model. And they said, you should remake it again. This is a quadruple transgenic mouse model. We would have sunk all our resources trying to make this mouse model because they wouldn't share it with us. So we stopped the study. We couldn't do it. Versus, I'm not going to share the cell line with you, or I'm not going to share that DNA plasmid with you. All right, we can do that, but it's just going to cost us more time and money to do that. Those are the kind of situations that we were running into. As a result of that, I had told you we had 36 at the end of design, and I'm about to slide into the very next stage. We didn't submit all of those. So we had 36 ready to submit to eLife for peer review. 4 of those we stopped. And we stopped because even though we got them that far, the writing was a bit on the wall. It was saying it's not worth sending it to the reviewers because there's no way we can afford to do this anymore because of all the mounting challenges we had. So then the question is, well, how did things go in peer review for at least the 32 that we sent, right? Because I told you 4 of them didn't go. Well, we lost another one because in the process of that, we realized that, again, the costs were too high for us. We did have 29 go through with revisions of varying degrees. And we had 2 rejected. So even though we had this format, we were trying to do our best. Peer review was still telling us, hey, no, that's still not worth you doing your research on. Maybe it's because the design wasn't good enough, or maybe they thought the experiments we picked weren't the right ones to pick. And that it was just, we couldn't come to a agreement on it, so we had to basically see them get rejected. And I'm happy to go into that more afterwards. Somebody can grab me if you want and have a beer and talk about it. There's a couple other things we're going to talk about too then, more beer. Okay, so let's move to the next stage. We're done, right? 29, they're going to hammer through. All right, let's stop again and figure out the top level. 29 we started, down to 89 experiments. I'm going to tell you that we didn't do all that. We're not finishing that. We're going to finish 23 papers, 52 experiments of varying degree. Only 18 did we actually do everything that we said we were going to do. This is after we designed it, right? 6 of the papers, we didn't do anything. We couldn't really get any outcome, right? The outcome is what I'm referring to. Some outcome to be able to compare back to the original study. 5, we had a mix, right? So maybe there were 5 experiments, but 2 of them we were able to do, and the other 3, they were sinks. We just couldn't figure it out. Even though getting all this information, we were expending our resources. And I'll give you an example of what I mean by that in a moment. All right, so modifications, it's essentially what I'm hitting at. So of the experiments that we had, these 29 that went through, 29 papers and the experiments, we had 76 experiments started. So we could assess, whether modifications were needed for those. There were 13 that, like I said, didn't get kind of really started, so I'm not going to include those. And so we see that, yes, we could, it's not showing. Why is it not showing? Sorry, I will read it to you. That we found that 67% of the experiments could be completely implemented. And that includes like enough there that basically we didn't have to do anything. They kind of did exactly like we thought they were going to. And that's 64% of the papers, so 18 of the papers were completely implemented, having an experiment in one of the papers completely implemented. What you're seeing here, because it's cutting off the last one, so if you go to OSF for the meeting page, you'll be able to see this. Please do that. We had varying degrees again, right? That we had to do some modification. And it varies a lot in terms of what we actually had to do. So let me tell you whether we did it, right? Because of course, I didn't just say I needed modifications. We actually tried. We were trying to do faithful replications to the best we could. And what we did is that we could, again, have completely implemented, again, that includes ones and the 6 papers, the 23 experiments for which no modification were needed, but we couldn't do it for all, right? We had varying degrees. So some of them we didn't implement. And again, this comes back to the resource constraint. It's not because we didn't try. We were trying to do this. It's just that basically we burned through the resources that we had to varying degrees. And we couldn't quite get over the hump of like, well, what's a proper outcome that you can get? All right, so so you can see this. Let me give you some examples. So I want to describe this. There's no pictures here. I figured we'll try this approach. Here's 2 papers, 2 different studies that we were replicating. As you can see, one of them I've published, so you can look that one up. The other one is not, so you can see where I'm going. So let's start with the one that we finished. So here's the experiment in particular that is a mouse model experiment. So it's a leukemic model. It's to inject a drug and be able to monitor the tumor growth in vivo using bioluminescence. So really nice. We're able to get everything that we needed, the cell lines, the tumor cells that had that bioluminescent marker, the drug formulation that we needed, the mice that we needed, all set to go, know what we're doing. Lab goes off and runs it. Around the right time that they think to, they start checking to see if they've got disease implementation, right? Basically seeing if it's established, because you need to check that first before you start doing drug treatment. Can't see anything, so we're like, oh shoot, all right, I guess keep waiting, take a little longer, and eventually get to the point where the disease manifests, right? And this is hind leg paralysis. So they see the disease progressing in the animal, but we cannot see any disease inside the animal in the sense of bioluminescence. So we're like really confused. We're like, we know that these cells have expression of this GFP marker, but we're not detecting it. So what do we do? All right, well, that's a shame, right? That this didn't work out the way we wanted to. We went back, we were able to contact the authors. They had some suggestions, basically looking for ways to really enrich the cells that were expressing the absolute most, doing a couple other different tweaks. We go back, run a pilot on a couple mice. We figure out, okay, well, we can do it. We just need to change the timeline, confirm that it's the appropriate change. This is increasing time and cost. Eventually, we implemented the change, and we're able to conduct the replication and get the outcome. In this case, it's actually a similar outcome to the original study, which is a good example to show. So that's one where we had really confronted with immense challenges, but we kind of took a step back, were able to work through it and re-implement it with this change. Again, being fully disclosed when we send this to eLife and for everybody to look at, and that they understand the justifications for it. Let's counter that with another one. So another mouse model. This one's looking at the impact of expressing an oncogene on the liver. So we're looking at some markers. So we're going to do some staining of the liver at the end of the study. But we have to inject this DNA into the mice. So again, we get everything from the authors. They give us the DNA plasmid. We know the mice. We begin the injections. And the mice start having seizures. And we have to shut the whole thing down because it's not ethical. Now we got to go backwards again. So we talk to the authors. We get new samples. We basically run more pellets. We figure it out. We move forward for a long experiment. Basically, the lab goes forward, finishing the experiment, excising the livers, doing the staining. All controls look great, but we're not seeing anything else anywhere else. So it's a whole bunch of zeros, essentially. So something's going on, right? So I guess I can show you a bunch of zeros, which we will when we publish this, but I can't really compare that because there's something else going on, right? Like I basically have a floor effect. But we don't know what to do. So this would suggest go back and figure out what's going on. Like your staining seems to work, but there's something still off in your approach. But we didn't continue that because that was incredibly costly, and we did these first hurdles to see if we could get through it. So these are just 2 examples, and there's varying degrees of other ones where we tried to move through it, and other ones we couldn't, even after sorting out all these details beforehand. All right, so you saw on the other slide that there's 18 that we fully finished. Of which there's a fraction more that are still going. So where do those sit? All right, so we've got one rejected. That's the beer. You can buy me again, and we'll talk about that one. I've got 3 more in revision. We expect those to be published. One thing to note here that I do want to do is I don't know if any of you have published in registered reports or done work in replications. Something that is interesting in this phase is the challenge of being confronted with that evidence as reviewers. So I'll give you an example of what I mean by that. So there's a particular paper we're looking at where they're looking at knockdown of a gene of interest and co-kitment, at the same time treatment of a drug. And the original study says, okay, if you do the drug by itself, you don't see an effect, but if you knock down the gene at the same time, we see an effect on cell viability. Okay, so that's a pretty nice study, very, very exciting result, groundbreaking, right, Nature paper. So we're like, cool, okay, we want to do that one. So we get this, we get all the information from the authors, it goes through peer review, everybody's like, perfect, you're doing the exact same thing as the original paper, everything looks good. We conduct it, and we see nothing. We don't see it, it looks no different than control. So we're like, hey, this is what we found. We did it the same way, everything checks out, this is what we found. And so then we had back and forths constantly trying to get this published, and the best reviewer comment that I have that kind of sums up what I'm trying to get at is a reviewer saying, well, I just don't know how that timeline that you have would have ever had an effect. Like your design is wrong, not recognizing that we didn't create the design. It's the original design, right? We're just mirroring that design. So then if the question is, we did it wrong because the timeline's wrong, how did the original get a result? But it's because we were confronted with ourselves, right? And the reviewers are challenged with that, which is, I don't like the result, it doesn't make sense, so I'm trying to figure this out. And I just find that one interesting because it's not our design question. All right. Let me give you a summary slide here of what we talked about, right? Started with 51, 197 experiments. We're way down to having a total of 52 experiments completely finished. We expect to have 17 accepted in eLife, and right now we're writing up a paper on this, so I definitely appreciate any feedback on this in terms of the directions that we can go and the way to communicate this. And naturally, for the ones that finished, we'll publish just like the psychology paper did. We'll compare those results. But I think it's important to remember how we got those results and the ones that we couldn't get as well. So I'll leave this up for a little bit in terms of some conclusions you can have. There's a number of factors I think that are in here. But in the meantime, because time's almost out, I'll take any questions you guys have. And thanks for your time.
Hi, sorry, I have asked a question before, but this is really a point of information. I don't understand how at the stage 2 of a register report, you're getting asked to do major revisions. I thought the whole idea at stage 2 was that it was basically accepted if you did what you said you were originally going to do.
But we did do what we said, but I didn't get the same result, Dorothy. So I've got to do revisions. That's the problem. So it got accepted. It got accepted, but that's the challenge we had, is that we basically would go sometimes back and forth multiple times. If anything, if it wasn't for the registry report format, if it wasn't for the fact that despite people trying to stop us, telling this paper should be rejected, we still got it accepted. So it is interesting. It's not that they did want more experiments, to be honest there, but we didn't have to do it. It's that there were these hoops that we had to jump through in presentation, the caveats. So many caveats.
But your
and I could
editor
never
was
do it.
not really then giving you a registered report, right?
What's that?
Well, the editor was not behaving like the editor of a registered report.
I think that a project like this and going into new areas confronts it. So I think you're right. There's room for improvement in the terms of how it could be implemented. But I think it's combined with the fact that they're unsure how to handle the disconnect between it. So the times it would go through really smoothly was when we got the same result, and the times that it didn't, it's very easy, I think, as an editor, because it is new to them, right? They did this for just this project, as they got confronted with, well, what do we do? And all the reviewers are telling us, especially the original author, saying we did something wrong and hunting left and right for it.
But the whole point is that it's independent of the results of the registered report.
We can talk about this more. I'm not disagreeing with you. We can talk about it more. This is another beer. Jeez, this is really fun.
First, amazing project, amazing commitment. I mean, I'm just blown away by this. In my own analyses of kind of approximate replications, so similar replications, people looking at the same finding, I find that if it's within the same author, within even the kind of extended author group, that it's much less likely that they'll find the contrary finding, for example, in a gene experiment, in a drug gene experiment, even in a clinical trial. Mm-hmm. So there's like massive underdispersion when you have a kind of high density author groups. And so I liked the idea that, hey, I'm willing to collaborate with authors, there are authors on these papers, but my guess is that the same experience you're finding with editors and reviewers, that when the findings are not the same but the protocols are the same, or even stronger with someone who's involved in the author group, who, for whom this is a meaningful part of the brand, who really feels or is committed to what it is that they did in the past. And I'm curious if that kind of like shakes out, if that is in fact the case, and how to think about the process of doing this. I see it actually as a fundamentally kind of oppositional activity. I don't think of it as a collaborative activity, because in the end, you don't actually want to produce like fragile, highly replicable effects. You want to, like for medicine, you actually want robust effects which are not precisely replicated in this space. So how do you think about I mean, it sounded like you were trying to kind of create a kumbaya, activity, and I just don't see how that's desirable, actually, in terms of the outcome for science.
Yeah, that's a great question. That's funny. I'm gonna think about me being like that. So the process that we were trying to do is, and I think this is really where, despite the fact that implementation issue is still, Dorothy rightly brought out, it's in the front end. It's when I'm designing it. So I think what we were looking for was saying, if I go out there saying, guns a-blazing, look what I found and how I did it, right, really fast. But I think if we say, I'm trying to do this, and letting them know, like, we're doing this because, by the way, somebody already told you that they couldn't replicate results, they didn't show you anything. We actually got, for the most part, pretty good response, or we got pure reluctance. They're gonna do it anyways, you better help them. Because we did it way in the early stage, like the attempt was to say, and I believe it still, right? My goal is to say, I want to get the same result as you. Tell me what to do. Tell the labs what to do, because we really want to find the same result. And whether it is like, and it's an interesting question to have, right? Which is, should I do exactly what you said in the paper versus should I do what you tell me? Is, we had this multiple times. I'd find stuff that was wrong in the paper, right? And they'd be like, oh, don't do that, that's a mistake, but we're not gonna fix it. But I'll tell you what we did. Because that's actually what we were hunting for. And that's what I meant by being collaborative at that stage, because if we're not, then the quick thing on the outset, which by the way, we still got anyways at the end, which is you did something wrong, you forgot this. And you're like, well, that would have been helpful if you told us in the beginning, and did you know that before we got our result? Your point being taken. But I do think you're right, like we did get a lot of times where original authors, and this is at least where the Rich Report helped us, they would try to tell the editors, you reject this, reject this. We've done it in multiple mouse models, right? How dare they just do it in the one? And again, our point was to say, I just want to pick a random set. Instead of just picking random experiments, I'll try to pick ones that are closer, but I should be able to pick up enough of them to be able to get some results. Even if it is, say, a robust result that maybe I just happened to find the one that was the false positive, for instance. But we can talk more about that. That's a really good question.
No, thanks.
So yeah, I should start by saying that literally was listening to it, and the image that came in my mind is this, Bruce Willis in Die Hard movies fighting all this stuff. So, and I'm saying it with any hint of sarcasm or anything. This is but because it's preceding, I would say, like a devil's advocate question. because when I was following your reports, and it's really close to my heart, and I think it should be all done. But at the same time, I just checked over the last 5 years, cancer research produced 488,000 something papers. If you search only on limit by studies focused on cancer, right? Not reviews, anything else. So the question then is, if you do this work, what do you think would be, and what kind of impact it can have, and what needs to be done to make this impact larger? How would you do it if you start over again, to put this question another way?
Yeah, no, there's a lot to go in there. I think one of the things that this work, because you are right, it's a drop in the bucket compared to everything that's going on. To me, I think it's more it's easy, going back to that first slide, right? It's very easy to say, I can't do something, I can't find it. I think it's more important to show what you had, what the challenges that you ran into. So what I hope this is a way to have us all here and not here think about what can we change in our current culture to try to basically help with this. And there's some low-hanging fruit here if you stop and think about it. And there's some great groups doing amazing things on it, which is how do you make it easier to share the protocol beforehand? Like we had that as the biggest challenge in here. Even talking to authors, it just all got lost, right? And there is this thought of like, doesn't it matter? Like, I'll give you an example of like one thing that I hope, and I think Fiona here helped publish that paper, which is sample size and biomedical research. Like I'm mad at myself, my younger self, because I imposed my belief when somebody said n equals 10, whatever it was. I was like, oh, I know what that means. That's what I was taught. That's what I'm doing. That's crap, just to use a clean word there, right? It's not what everybody does, right? I made an assumption, and it's the wrong one. So I think we need more of this to just try to give more evidence to the changes that people are trying to do versus just saying, let's do it because it feels good. There's the anti-kumbaya. And that's what I think that's one of the things. The other thing is, I think, we'll go out to using pharma as an example, which is right now we know that there's a problem translating drugs and translating anything from preclinical to clinical, right? There's the big bridge, phase 2, things tank. And I'm not disagreeing with one of the big factors there, which is we don't understand fundamental biology to be able to make that bridge. Yes, we can help mice. Mice are not humans. And there's ways that you can think about a way to do that. I think there's one easy way to do that, which is, are you sure that result even was worth the time going to a clinical trial? Because if you're just scattering around picking experiments that make your case, I think you're doing a disservice. Now, this project can't get on that, but like, I'm more than glad to talk to you about ones I think that maybe Janssen and other companies could do to try to tackle that a little bit.
Thank you.
Yes, well, great lecture. It's great to see this coming together. It's much more fun to see the whole thing. Question goes again to the registry report format. We might need a few beers for that. I do agree that maybe the editor is not behaving or the reviewer is not behaving. That said, isn't there something about the type of research? I mean, I think we came up with registry reports much based on clinical trials, which had like a very standardized protocol that's followed like 2, 3, 4 years. Psychology has big experiments as well. I mean, basic lab science is usually like 50 experiments, each of which you're tinkering and trying and increasing the dose and changing the cells and whatever. I mean, can that have to do like the limit of the format? I mean, do you think registry reports, as we know them, are actually amenable to this kind of like complicated lab science, or
Yeah.
do we have to think of other ways to pre-register and yeah?
I'll give you. I do think it is. But I get your point, and I agree with it. I'll give you an example for anyone that's familiar with the ARRIVE guidelines. So the ARRIVE guidelines are reporting guidelines when you do animal research, and they're reporting guidelines. So right now the emphasis tends to hang out on the back end, which is what did you do among these really important criteria, by the way, really, really key ones to make animal research the best it can be. Because if you're not going to make it the best, don't bother doing it. That's the wrong place, in my opinion. That deserves to be up front. That's what a registry reports helps you do. You use these norms that have already established for techniques. What are the details that are the most pressing that one should do? And tell me what those are. And the easiest way, I think, to do it is to prompt a little bit, which is, I think, what we can help a little bit, which is don't just make it be a free-for-all. And again, we did it a little bit naively because we were just following the best that we could. But in hindsight, I would do that, which I know that you're thinking of doing as well, right? Which is take standardized formats to make sure that the questions are being asked, so that way it's easier for people to be able to review across them. And I think ARRIVE guidelines is a great example to point out.
Thank you very much for the talk. Amazing. One recommendation that comes to mind, listening to your talk, it's that as a low-hanging fruit, it's basically upon submission to a journal, researchers just should, as a line item, they should just say how much would it cost to reproduce a paper or a result. Is that possible? Is it sensible? What are your thoughts about that?
Yeah, no, that's an interesting question. I would vote more for what am I making open to allow my research to be reproduced. But I like that point where you're going. This was an interesting project. When you think of, again, any other typical research, you don't break it down quite this way, right? You're like, here's a couple $1 million. Go do the things that you said you're going to do. Show me the papers you got at the end. It's really interesting to kind of break it apart this way, to have a question. And again, this is not, there's more costs that were there. We kind of got reduced rates a little bit. We had companies giving us helps with reagents, for instance. So this cost is actually higher. And I think it's because it's hard to pin down what it really is. And I don't think it's really, in light of this data, I don't think I could still even project properly forward what we'd have, because I think we've not had enough sample to actually properly give a good estimate. But it's an interesting thing to think about. Thanks.
So early on in the process, when you were reaching out for either data details or protocol clarifications, for the people who wouldn't or people who didn't, were you finding it was more a, was it a can't or a won't situation, right? Were you running into essentially bad record keeping where they couldn't give you the details and say, sorry, it just doesn't exist? Or was it confidentiality agreements or intransigence or what? Like, what
Yeah, that's a really good question. Almost entirely, I can't do it. And it is like literally like the dog ate my data. In this case, it was the postdoc. The postdoc seemed to really mess everything up, but that's why I didn't do a postdoc, Ryan. Yeah, it was they can't, and it's really amazing to hear the challenges, which is basically like, oh yeah, I don't know where the data are. Geez, this would take me so much time to do this. I don't know if I can even figure it out. I don't know where the files are, and it is literally like, oh, since I moved, like yeah, they wiped the computer that it was on. So and it's because there's just it's not the norm to say, oh right, I can make that public somewhere, right? And that I think has changed. So the vast majority of it was, I just can't do it. The only time we had a won't was somebody who basically was quite interesting. It was just they opted out entirely. Like, nope, I like what you guys are doing. Do it on your own. It's in the paper. It's like, all right. Which according to eLife did not replicate. So that's something else to think about.
So Tim, there was a paper, I thought it was a bit of a hatchet job, or something like that in Science that was talking about essentially the inability of the editors to hold people to proper standards at phase 2 of the review. And I'm just curious, without going into the details of that, I'm curious, what was your experience? Did you feel like you received adequate pushback on changes to protocol and the likes when you went through the second round, or did you feel like people didn't know what to do with it and were just like, okay, yeah, sure?
No, totally that other one. Like we got a lot of pushback, and it was really of this light of, okay, I see you did that, but really, like, you didn't quite do it right. And sometimes, and like, I'm not going to also masquerade it was all like that. It's a great example of one where we made a mistake and didn't know about it until stage 2. So it was the timeline was, again, arrive guidelines. We should have done that. We should have used a figure right in the beginning. The paper didn't have any clear figure of like what the timeline was. It was very confusing. We mirrored it. Again, we shared it with the authors, peer reviewed it. It was public, published openly. Everybody could look at it. We do the results, ran into all these challenges, published the results. We're like, geez, okay, this is the best we could do. Like, we just couldn't figure this one out. And I remember reviewer 1, 2, 3 is like, yeah, they really tried hard to do this. And reviewer 4 is like, you screwed it up. You did it wrong. The timeline's wrong. And I was like, wait, what are they talking about? And it must have been the original. It's the only way it's the original author, right? Because they actually used themselves in the third person. But anyways, we did it wrong. We did it wrong, massively wrong. We made the wrong assumption. And the editor in that case was like, you're right. Like, the original paper is horrible. Like, nobody knows what happened. So in that case, we had good forgiveness, but like that's the one time where like they were right. We got it wrong, like, and it upsets me a lot still because we wasted a lot of resources and a lot of animal lives. That, geez, if we had just known how to do it the right way. So sometimes it was for the right reason, but a lot of times it was just like grabbing at straws. It's actually, to be honest, in my experience, I'd be curious about others. We make everything open to the peer reviewers, the entire process. Man, they go digging left and right. They're like, you didn't pipette right, you didn't do this right. And you're like, well, you only know half of this because I showed it to you. So it's a lot easier for you to criticize my methods. I have no idea what you did to this level of degree. So we got a lot of that. And I think it's grappling with like, oh, I don't like this result, and I'm trying to figure out how I can justify it after the fact. Yeah.
So is this really a problem with our current publishing system and the way we publish work? The fact that there are gatekeepers, either editors or reviewers, and should we be thinking about different ways to communicate science that doesn't involve these gatekeepers, makes sure that much of the data and the protocols are available? What are your thoughts on that? If you had a magic wand, how would you fix this?
If I had a magic wand, how would I fix this? I don't think journals in their current format help, for sure. But I think the one that I'd rather see is that, and actually, in light of the challenges that we had, is the fact that I don't think it's right to have one's career hinge on an outcome. So like, if that's changing journals, that's great, but it's gotta change the way we fund, it's gotta change the way we hire and the way that we promote and judge people's careers. So that magic wand is, gotta cover all that, in my opinion, because I feel if you change one, you don't change the rest, you're just gonna, the system doesn't really move, but that's a really good one.
This will be your last
Okay.
Thank you, Tim, for remarkable and heroic effort, really, on this project. You also generated a very interesting data set to really look at something and give visibility into something that happens every day in the labs. People are trying to replicate every day. My question is, I mean, a lot of the difficulties you ran into could have been avoided if methods of sharing more openly and reporting, following the ARRIVE guideline, following good reporting guideline, making your data available, all of that is possible today. Do you have a sense of how much easier it would have been for you to do that if all the papers you tried to replicate had available data, code, protocols, reagents identified unambiguously shared, and so on?
Yeah. It'd be, and right, and I'm glad you said it. Like, I would want materials, right, like repositories. Great, AdGene's a good one for those who know it. There's a lot of great, like, physical repositories out there. Yeah, if everything was done in a manner that we could access, and all we truly were talking about were the finer details, the nuance, that even though, we can capture that, you could argue videos is a great way to do it as well. I think our cost would have been still gone up because things happen. Things just happen. But I think the slog, I think somebody used that word and it's the right word, that we had to go through to try to figure, and a lot of it is just trying to understand what was done so that way we're doing the best that we can. That would have been minimalized. So I'm sure, just like anything else, more things would open up. It's not like everything would be rosy. But I think it would have allowed us actually probably get a little closer to the research questions versus I think where we got hung up, not to like minimize it, which is we couldn't even get there sometimes because we had so many technical barriers accessing information, understanding information, that, it just seems to be, it's not set up. That's the right way to think about it. So, all right, thank you all.